Epnot, the golf ball shaped wonder of the world that's brought intrigue, delight, and nucleic acid knowledge to generations. But like anything imbued with greatness, Epnot hides a dark secret. Welcome to the train graveyard, where old, broken, mutated rides go to die. And what better place to talk about DNA mutations? Mutations result from damage to DNA, and there are a scarily large number of things that can do some damage. First, the exogenous sources. These are external agents like UV light, ionizing radiation, cigarette smoke, asbestos, and intercalating agents, which insert themselves between the DNA base pairs. And this is why your parents told you to never smoke. But down here in the train graveyard, down here are the bad kids, sneaking off and making bad decisions. And then there's the endogenous sources, like reactive oxygen species and alkylating agents that can wreak havoc on nitrogenous bases. They arise from normal everyday metabolism. Lastly, DNA polymerase and proofreading errors are a source of DNA damage. Scratchy land? Man, someone should have proofread that. But it does kind of fit. It's pretty scratchy down here. Fortunately, our bodies have different repair pathways to identify and fix damaged DNA. But we'll save those details for another sketch. Alright, so now that you know that DNA damage can occur from basically everywhere, here are the various DNA mutations. We'll begin with the single nucleotide, or point mutations, divided into transitions and transversions. A transition mutation occurs when a one-ring pyrimidine replaces another pyrimidine, or when a two-ring purine replaces another purine. To remember that, we've illustrated this track transition. Note the pyramid cart replacing another falling pyramid cart. At Sketchy, the pyramid is our recurring symbol for pyrimidines, but you probably already knew that. In contrast, a transversion mutation is when a purine replaces a pyrimidine, or vice versa. Shown here is a prankster dressed in a nun costume humming a verse and pushing a pyramid cart. Nuns are pure, like purines, but I'm gonna wager the only thing pure about this rapscallion is his love for mischief. Transitions and transversions are big overall categories that can be further subdivided into silent, missense, or nonsense mutations. A silent mutation happens when a base substitution produces the same amino acid. It's called silent because you wouldn't be able to tell there was a mutation just by looking at the protein sequence. Ah, Brayden, why can't you keep your hands to yourself? Now give back the stuffed animal and don't silence your brother. Silent mutations often occur in the wobble position. To learn more, wobble on over to the RNA structure and function sketch. Okay, next is the missense mutation. Here, a base substitution produces a different amino acid, and therefore a different protein. An example of a missense mutation is sickle cell disease, represented by the sickle, of course. In sickle cell disease, valine replaces glutamate in the beta hemoglobin gene, leading to sickled RBCs. Alright, we'll finish up this section by going over the nonsense mutation. This happens when a base change results in a premature stop codon. Nonsense mutations are much worse than silent and missense mutations because they produce truncated, non-functional proteins. This mutation is represented by a maintenance worker pushing someone off the ride. Look, sometimes you just get tired of these whippersnappers messing around. Note the red lantern and falling stuffed turkey, because we're replacing an amino acid, in this case tryptophan, with a stop codon. Time to change gears and discuss frame shift mutations, which disrupt the reading frame. They're caused by adding or subtracting any number of nucleotides not divisible by 3. Why is this important? Well, remember that 3 nucleotides make up a single codon and code for a given amino acid. Adding one nucleotide, for example, will change all the downstream codons, and possibly all the amino acids, resulting in a completely altered and usually non-functional protein. Hey, they've got 4 in that cart. They're only supposed to have 3. This is a park with rules, people. That multi-cart pileup down the track will remind you that frameshift mutations cause significant downstream errors in the resultant polypeptide. So, what are some genetic disorders commonly caused by frameshift mutations? Well, here's a kid in a Scottish tartan. No, being Scottish isn't the mutation. A Scottish tartan is the usual garb of the Duchenne clan, and our symbol for Duchenne muscular dystrophy. And what's he carrying? Looks like a whole frickin' sack of stolen track parts. Just another day at Scrutchyland, riding the rides and doing the crimes. This sac is our symbol for Tay-Sachs disease, another genetic disorder resulting from a frameshift mutation. So here's a summary of all the point mutations we've covered so far. Now, the last thing we'll cover are splice site mutations. They occur in donor or acceptor splice sites of introns, or non coding regions, just adjacent to exons. As a reminder, introns are spliced out of eukaryotic pre messenger RNA, while exons are spliced together to generate the final mature mRNA. Exons are expressed, as the saying goes. At least that's how it's supposed to work. But splice site mutations may result in either incorporation of introns or exclusion of exons in mature messenger RNA, leading to an altered and often non functional protein. So, now that we've covered these mutations, you might be wondering, what's the big deal? Well, mutations can cause lots of different problems. But one particularly scary one is that unchecked DNA mutations can cause cancer if specific genes are damaged. Proto-oncogenes normally encode for cell proliferation proteins. They can undergo mutations to become oncogenes. When expressed at high levels, oncogenes allow for cells to grow and proliferate, accelerating through the cell cycle at rapid speeds, eventually leading to cancer development. Here's a visual representation. Imagine not being able to take your foot off the throttle, kind of like a Keanu Reeves movie. That's basically how oncogenes work at the cellular level. On the other hand, tumor suppressor genes, like p53, encode for proteins that prevent cells from progressing through the cell cycle, through downregulation and activating DNA repair. Interestingly, these functions persist until both alleles have been mutated. This broken break will remind you that reduced expression of tumor suppressor genes allows cells to pass through the cell cycle without any checkpoints or breaks, eventually leading to cancer development. Look out below! As fun as it is to do bad things, it's getting a little smoky down here. Time to head back up above ground. UV light, ionizing radiation, cigarette smoke, asbestos, reactive oxygen species, alkylating agents, and proofreading errors are the major sources of DNA damage. Nucleotide substitutions are classified as transitions or transversions, and further divided into silent, missense, and nonsense mutations. Silent mutations occur when a base substitution produces the same amino acid. Missense mutations occur when a base substitution produces a different amino acid, like in sickle cell disease. Nonsense mutations occur when a base substitution produces a premature stop codon and truncated protein. Frameshift mutations are caused by adding or deleting any number of nucleotides not divisible by 3. Usually, this results in a completely altered and often non-functional protein. Examples include Tay-Sachs disease and Duchenne muscular dystrophy. Splice mutations occur in donor or acceptor splice sites, resulting in either incorporation of introns or exclusion of exons in mature messenger RNA, leading to an altered and usually non-functional protein. And finally, overexpression of oncogenes and underexpression of tumor suppressor genes can result in cancer.